Okay, let's talk about osmosis. So if I see the word osmosis, water is the only thing that I'm thinking about. If I have a cell and I put it in an environment where there is a lot of water, okay, what's going to happen to that cell? Well, the water always moves, water always flows from where there's more water to where there's less water, okay? So water is always going from high to low concentration. It's going down the concentration gradient, which means it's passive. It does not need energy. So when I look at this diagram, if this is my cell, water is going to go from where there's more water to where there's less water. What will the cell look like when it's done? Well, if you're absorbing water, what's happened to the cell? The cell will expand, okay? Expand. It will get bigger. If it gets too big, it will burst, okay? This is when we have more water to less water. This is called hypotonic. I'm not sure that's a terribly important word for you to know, but all you do need to know is water moves where there's more of it to where there's less of it. Now, if I put a cell in an environment where there's more water inside the cell than there is outside the cell, again, water always moves from where there's more to where there's less. Well, now there's more water inside the cell, so this water is going to diffuse out of the cell. What is the cell going to do now? It's going to shrink, okay? It will get smaller until it's completely collapsed and can no longer function as a cell. Osmosis, water, from where there's more water to where there's less water. Okay, looking at this one here, if you start with one cell and you end with 4 cells, you are talking about meiosis. Okay, so this is meiosis, and in meiosis, first off, we have to recognize the importance of meiosis. And so the importance of meiosis is threefold. First, it produces gametes, sex cells. The second reason it's important is that when it divides, we go from pairs of chromosomes, which make copies of themselves, and then this goes into here, this one goes into here, this one goes to here, this one goes to here. So we go from the chromosomes being in pairs or diploid to the chromosomes being singles or haploid. See, these are just singles. So we reduce the chromosome number. And then the third reason is down here, because if you recall the chromosomes, this is the chromosomes from mum, this is the chromosomes from dad that you have, and during prophase 1, they do this. They kind of trade out bits. That's called crossing over. And crossing over produces genetic variation or genetic diversity. It's why you look like your siblings, but you're not identical. So if you see a diagram where you start off with one cell and you end up with 1, 2, 3, 4, this is meiosis. It produces gametes. We go from diploid to haploid, and we get crossing over, which gives us the variation. Okay, moving on up to this part, protein synthesis. We talked a lot about this and about why, what our DNA actually codes for. So when we're talking about protein synthesis, step number 1 is the DNA opens up and becomes a template to make messenger RNA. Okay, that's step number 1. because we're talking about DNA, it's got to happen in the nucleus. The next stage is the messenger RNA leaves the nucleus, all right, and it goes to a kind of a structure that we talked about earlier called the ribosome. And in that, we use the messenger RNA to assemble amino acids together in a specific order. And amino acids, as we know, form the building blocks of proteins. And so if you have a sequence of messenger RNA, how do I know this is RNA? Because I've got to use step number 1. Always remember, break it down into 3s. And then you're going to work out which amino acid this is by using our codon chart. So if you look here, I'm just going to move this up a little bit. Our first one is AUG. So we go first letter A, second letter is U, so it's going to be in this box. G is over here, so it's going to be methionine, M-E-T. The next one is A-C-C. A, first letter, second letter C, third letter C is threonine, so that's T-H-R. And then the last one, G-U-A, first letter G, second letter U, A, that's going to be valine. Okay? If you see a codon chart in the question, that means you have to use it to answer the question. Okay? So that's how we put together amino acids in a certain order to make a sequence of amino acids that build up a protein. Now, here's the next bit. What happens if we change the sequence of nucleotides? That is a mutation. Okay? And sometimes they are what we call silent, or they don't change the amino acids because they code for the same amino acid. For example, if you look here, you can see how C, U, this one codes for leucine, but CUC also codes for leucine. So if I change and CUA codes for leucine, and you can see they kind of like all these different things code for leucine. UUG codes for leucine. So if I change the order of the nitrogen bases in messenger RNA, because I've changed them in DNA in the first place, I may code for the same amino acid, in which case we call it a silent one. But remember, if let's say AUG is our proper one, all right, that's the normal. I know if I look at it and it says the mutated one is this, I know that I have an insertion. Why do I know? I've added something because this is longer than the original one. If my was just that, I know this is a deletion because it's shorter. So it's easy to spot an insertion and a deletion. If everything's messed up, remember that's called a frame shift. And if it's the same thing, so like this one, A, G, G. Okay, so I'm looking here. This is the mistake. This is called a substitution or a point mutation. If it's longer, like we've added an extra G, that's an insertion. And if when you count it, it's shorter, that's a deletion. So any mutation is going to be a change in the nitrogen bases. Sometimes it will code for the same amino acid and you wouldn't even know that there was a mutation there. Okay, on this last part here on the front page, we're talking about gene expression. Gene expression is taking a section of DNA, right, and converting it into a protein, and that protein gives you a characteristic. That's one, or a trait. Okay. Another thing about gene expression is this. Let's say we have an entire strand of DNA, and that same DNA is in every cell. So why is it that some cells are muscle cells and some cells are nerve cells, and we got all these different types of cells? And the reason for that is that you start off with a basic cell, and if this section of DNA here is turned on, that basic cell may turn into a really long nerve cell, right? Because this section is turned on, but this section is turned off. But if this section of the DNA here is switched on, right, you may end up with a muscle cell. So every cell in your body where you have a nucleus has a complete set of DNA, but only certain sections are expressed in any given cell. So this cell here, this section here, expresses the characteristics that make a cell a muscle cell. This section here, the characteristics expressed would turn the cell into a nerve cell. And that's what we call gene expression. If you see this phrase, particularly regulation of gene expression, then that's a pick me that's almost always the correct answer.